Hello everyone and welcome to Kognito Key. I am absolutely thrilled to have you all here today. We're going to be diving into something that is just wildly fascinating. We're talking about the ultimate survival capsules of the microscopic world. Yep, we're looking at the secret life of spores.
Picture this for a second. You are facing literally the most brutal, unforgiving conditions on planet Earth. We're talking boiling heat, massive chemical exposure, or maybe even decades without a single drop of water. Now, if you're a microscopic organism, how on earth do you survive that? How do you keep your entire species from just being completely wiped out? Well, it turns out evolution came up with a pretty brilliant workaround.
And that workaround is the spore. Basically, a spore is this incredibly tough, single-celled reproductive structure. When they're created through a specialized cell division, they actually only carry half of the parent's genetic material. So when things get rough, when nutrients totally dry up or the environment becomes toxic, certain bacteria and fungi form these spores. They basically pack up their essential genetics and go into this heavily armored lockdown just to endure the absolute worst. Okay, let's dive into how these are categorized. Spores are broadly classified into two distinct types, and it's based entirely on where they physically form in relation to their parent cell, which we call the vegetative cell. On one side, we have endospores. Endo literally means inside, so these guys form completely inside the parent cell. And on the flip side, we have exospores, which, as you might guess, form on the outside. Let's actually peel back the layers of this thing. It's really like a biological suit of armor. First up, you've got the outer and inner coats, made of these really dense proteins. They act as your frontline defense against chemical and enzymatic attacks. Just beneath that is the cortex. That's a thick peptidoglycan shield, and it's absolutely crucial for fighting off dehydration and super high heat. Then, moving inward, we hit the germ wall, giving us yet another layer of chemical protection. And finally, right there in the middle is the central core. That's the safe room, holding the precious DNA and RNA. Now here's a stat that's just mind-blowing. Inside that central core we just looked at, up to 40% of its structural components can be made up of something called dipicolinic acid, or DPA. This specific chemical adaptation is literally designed for ultimate survival. It stabilizes the genetic material so the spore can survive extreme heat that would, without a doubt, instantly destroy a regular cell.
Phase 1, sporogenesis, the 8 to 9 hour biological deployment.
So when a normal vegetative cell realizes it's in grave danger, it kicks off this incredible microscopic construction project. The whole thing takes about 8 to 9 hours, and the sequence is super organized. Step 1, the cell replicates its DNA and lines it up perfectly. Step 2, the membrane folds inward, making what we call a forespore. Step 3 is kind of sad but amazing. The original parent DNA actually disintegrates. It's literally sacrificing itself. Step 4, that heavy cortex and spore coat we talked about get deposited. And finally, Step 5, the fully armored endospore matures and gets released out into the wild. Now, to contrast that, let's look at exospore formation. Instead of building from the inside out, exospores form right on the outside surface. The vegetative cell undergoes cytoplasmic division, and this little outward bud starts to develop. This bud grows, it forms its own double-layered membrane and all those protective coats, and then it matures before finally popping off and detaching from the parent.
Phase 2, dormancy, the dramatic metabolic standoff.
You really shouldn't think of dormancy as just some simple, relaxing nap. It's a dramatic standoff. The spore shuts down entirely, becoming biologically inert just to wait out the storm. And this suspended state actually falls into two main categories. You've got endogenous dormancy, which is caused by internal biological locks, like a metabolic block or preventing water from getting in. Exogenous dormancy, however, is forced on the spore by external threats. Think extreme temperature swings totally drying out, or really harsh pH levels.
Phase 3, germination, the return to life.
So what happens when the environment finally gets friendly again? When the water returns and the temperatures normalize, the biological bunker opens its doors. This return to a vegetative state happens in a really precise sequence. Step 1 is activation. The dormant spore essentially needs a wake-up call. This gets triggered by a specific physical or chemical shock, like a sudden jolt of heat, some physical scraping, or low pH. Step 2 is initiation, and Step 3 is outgrowth. Let's actually zoom in on that middle step, initiation, because this is where the spore physically breaks down its own armor. The spore rapidly starts swelling up with water.
Then that incredibly tough protective spore coat finally ruptures. And because it's shedding its armor, the spore suddenly loses all that amazing resistance to environmental stress we've been talking about. The internal components are released, and metabolic activity just massively spikes. This perfectly sets the stage for the final outgrowth step, where the cell absorbs those essential nutrients and gets ready to divide.
From an impenetrable biological bunker right back to a thriving, dividing cell. It really makes you wonder, right? If microscopic life can engineer these ultimate survival capsules, what on earth could humanity learn from them for our own survival, maybe even out in the extremes of space? Thank you so much for joining me today on this journey. Stay curious. And please remember to subscribe to the Kognito Key channel so you never miss out on our deep dives. If you found this explainer helpful, please go ahead and hit that like button and share it with your friends. I will see you all in the next one.